so far?
Here's my computer is working.
Change the name there. What we're going to do today is a little bit of Punnett Square. You can all see that where it says Punnett Square there, I assume. Good, good. So yeah, so we're going to do a little bit of review of Punnett squares, and then we're going to talk about DNA and RNA and how they are made, how they work. I want to go into a little bit more detail than your book does, and hopefully explain it. I mean, this happens. Remember, this happens like a billion times in your body every day. So we need to make sure that we understand exactly what's happening. All right. So this guy, you learned about him in your lesson last week. One of the lessons. What's his name? Ooh, someone remembers his name. Hey.
Gregor Mendel, I think.
Very good, very good. Yeah. And he worked with peas. So he was the first scientist to really start researching genetics. So we call him the father of modern genetics. Now, when you read about him and the more you learn about him, the more you know that a lot of what he thought was right was later proven to be wrong, but a lot of it was right. Enough of it was right that he went down in history. He's an important scientist for us to remember. And one really important thing that he did is he used very good lab procedures. So he took meticulous notes. He worked with very large sample sizes, which of course he could do because they were peas. And peas were a big part of the reason that he was so successful. They're really perfect for this kind of study. Think about the difference between peas and, say, people. You know, peas have a very short life cycle as opposed to humans. Peas are a lot easier to manage in a lab. People don't really like to be locked up in laboratories and experimented on. Peas don't mind. Peas have lots of offspring, so it doesn't take very long to have a lot of peas that came from the same parent. That's not true of mammals and certainly not of people. So the work that he did led us to what we know about genetics now, and this is just really a huge field. Genetics is the major direction, I would say. Genetics and biotech is where we're really going with science these days. So I want to talk a little bit about Punnett squares. You had them last week. You would have learned about them, but I want to make sure that you understand them, not just for this unit, but for the next couple of units as well. So we need to make sure that we understand a few terms, that we're all on the same page. So this, of course, chart is 4 squares there, is called a Punnett square, and we can use it to predict the possible genotypes and phenotypes of offspring when we know the parents. So what do those terms mean? What is a genotype? Let me remind me. What is a genotype? Toby.
It's like the organism's like makeup of like a trait.
Good. It would be the alleles that it has, the actual genes that it has that give us the trait. So then what do we call the trait? If the genotype is the alleles that give us the trait, what's the trait itself? Kate.
Phenotype, I think.
Yeah, that's the phenotype. Excellent, excellent. So here you can see them. The phenotype is the physical appearance of an organism. I put what it looks like in parentheses there because there's a whole lot more to it than than the way it looks. Like for instance, your blood type is a phenotype, and it doesn't look like anything. You know, it doesn't look different than anything else. But a phenotype is the physical manifestation of the genotype. And of course, when we're talking about genetics, we also talk about the relationship between two or more different things. So how many green peas in relation to the yellow peas? How many wrinkled pods in relation to the smooth pods? Those kinds of things. We use ratios in order to figure out. All right, a few more terms here now. Alleles. So you've got this long stretch of DNA, and it twists itself up into those chromosomes. A piece of the DNA that codes for a protein is called a gene. You get two different of the same gene. You get one from your mom and one from your dad. So two different forms of those genes. Those are called alleles. So like the gene that gave you the color hair that you have, you got one allele from your mom and one allele from your dad. They're both the same gene, but they're different forms of that. You might have gotten brown hair from your dad and blonde hair from your mom. And you end up getting the brown hair from your dad because brown is dominant over the blonde, right? So when we're talking about alleles, remember, since you inherit one from each parent, you have two alleles for every single one of your traits. So you could have two that are the same. You might have gotten an uppercase T and an uppercase T from both parents. That would be homozygous dominant. You might have gotten a lowercase t and a lowercase t from both parents. That's homozygous recessive. If you got one from one parent and then the other from the other, so one dominant, one recessive, that would be called a heterozygous trait or heterozygous organism. Right. A couple more terms. Dominant and recessive are important ones. So remember, dominant shows up even if there's only one of them. If you get one dominant trait from one parent, doesn't matter what the other one is, that dominant one's going to show up. If you get one recessive trait from a parent, if the other one's dominant, the recessive isn't going to show. The only way for a recessive to show is if there's no dominant around. You have to have two recessive alleles in order for that recessive trait to show up. Now we can use Punnett squares with all this information. We can use it in order to predict the possibility that you're going to get a certain genotype, a certain, two genes from your parents. And then of course the phenotype that goes along with them. So we're going to make a couple of them, and we're going to talk about how we evaluate them. And then, we'll see how we use them. And hopefully this is review. I hope you guys have all gotten to that. We're not going to spend a lot of time on this, but it is important. So I want to make sure that you know them. All right. So what would go in this square right here? The way I'll talk you through the first one. Go ahead, Kate.
Did you ask what would go there?
Yeah.
It would be capitalized B, lowercase b.
Good. I often will say big B, little b, but capital is a little more formal, a little more accurate, I think. Yeah, so the way we do this is we drop them down. So we drop the B's down from above into every one of the squares. And then we bring the one from this side over and over. So Kate's right, that first one would be big B, little b. In fact, all of them would be big B, little b. So when we're looking at this, if B, big B, this one, is the dominant allele for the long beak phenotype, okay, we're talking about an animal with a beak here. The long beak phenotype is the big B, and little b is the recessive allele for the short beak phenotype. We can make predictions about the outcome here. So our phenotype, what it looks like, is going to be 100% long beak because they all have at least one of the dominant alleles. So we're going to have a 4 to 0 ratio, long beak to short beak. And our genotype is also going to be 100%. They're the same, right? Now, we can go backwards with this. If we know what the children are likely to be, we can figure out what the parents were. So in this one, the way we're going to do this is figure out what went here. Nope, wrong one. So let's say this B right here, that's going to come from that parent right there. then this B has to come from this one. And yep, that works because this B can come from this one, and then this one is going to be there too. So we know that one of our parents is homozygous dominant. Now, since this big B and this big B here are going to give us this one and this one, we know that we have to have a lowercase b right here. So we use the Punnett square to go backwards. So we know that our genotypes of our parents is homozygous dominant and heterozygous. Two big B's or big B, little b. Now things get a little bit tricky when we're talking about the male and female phenotype. So because male and female don't have a dominant gene, it isn't like one is dominant over the other. You're going to get two and they're not dominant. One isn't dominant. If you have one X and one Y, you are male. If you have two X's, you are female. So let's do the cross here. So what's going to go in this first box right here? Somebody tell me what goes there. Reagan.
XX.
Yep, you're going to have two X's. How about the next one right here? Kate.
XX.
Yep, and here? Stella.
XY.
XY, and finally, here's the last one, Kate.
XY.
XY. So looking at that, what are the odds of getting a female child? What is the likelihood that we will have a female child with this cross? Kate,
50% chance.
50%. And remember, every single time it's 50%. So these parents, this female and this male, could have 15 children and all 15 of them could be female. Or all 15 of them could be male. That's possible. Just like with that guinea pig question, there's a 50-50 likelihood. Now, really, the odds of flipping a coin 15 times and all 15 times having it land on heads are not great, but it's possible, right? And luck of the draw can have it happen that way. So every single time there is a fertilization, there is a 50-50 likelihood of getting female or male. All right. Anybody have any questions about this Punnett squares, the way we do them, what we can use them for? You feel like you're good with that? Okay. All right. Going on. All right. So this week, you're going to be learning about DNA and RNA. And I think this is one of the more fascinating topics in all of biology. I always picture in my mind when I'm talking about these nucleic acids and the way DNA and RNA do what they do, replicate themselves or make protein, is to think of a jacket that has a zipper on it. So think of your zipper on your jacket and how the teeth kind of line up with one another. And they have to be in the right order in order for the zipper to be able to zip up. If I damaged one side of my zipper, I could use the fixed side in order to, the undamaged side, in order to build a new one that was identical. That's a lot the way DNA and RNA work. So hopefully if we talk about it today, it'll make a lot more sense than when you just read about it. So that's what we're going to do for the rest of today. We're going to get as far as we can, and then we may have to pick up next week to finish it up. So remember, there's 4 kinds of what we call macromolecules in living things. Those big molecules, the big major ones, what are they? 4 macromolecules. I'll give you a hint, nucleic acid is one of them. What are the other 3? Toby?
Amino acids.
Amino acids make up one of them. What do you get when you put a bunch of amino acids together?
Proteins.
Yeah, protein. So protein, amino acids would be a micromolecule. The macromolecule would be the protein made up of those amino acids. So yes, you're on the right track. So we've got protein, we've got nucleic acid. What are the other 2? Macromolecules. Hey.
Is carbohydrates one of them?
Carbohydrate is one of them, yes. Carbohydrates are sugars and starches. They're made up of those monomers to make a polymer. So we got one more. Fatty acids. Sometimes you can just see them called lipids or fats. They all have a little slightly different meaning, but for our purposes, protein, carbohydrate, fat, nucleic acid, or fatty acids, let's say, and nucleic acid. So DNA and RNA are nucleic acids. Where do you find the DNA in a eukaryotic organism? Remember those two important terms, eukaryotic and prokaryotic? Kate?
In the nucleus.
In the nucleus, yeah. So does a prokaryotic organism have a nucleus? No. Does it have DNA? Yes. It has DNA in just a space. It's just an area of the cell that we call the nucleoid. But it's still DNA. They have to have DNA in order to make protein, right? So the nucleic acids that we're going to be dealing with, now there's 3 major nucleic acids. There's DNA, RNA, and guess what? ATP is a nucleic acid. But for now, the ones that are involved in genetics, we're talking DNA and RNA. Now, here we are at the cell cycle again. This is really important when we start talking about DNA and RNA and genetics, which is where we are now. So you guys remember the phases. Let's go through the phases again. That big one, A, that's like the waiting room in the cell cycle before mitosis begins. That is called interphase. So interphase is the longest phase. There's a lot happening in interphase though. There's that B right there. That's the growth phase. In that first growth phase, the cell is growing. It's duplicating a lot of the things, and especially the proteins that it's going to need in order to go through the cell cycle. Then we have C right here. That is the S phase, S for synthesis. That's where the DNA is duplicated. And it has to be an exact copy of the DNA before. This is the phase that we're going to be talking about today, is this DNA synthesis phase. Then we have the second growth phase, and we've got checkpoints in there. Remember, we talked about cancer if the checkpoints go wrong. Then we have mitosis. So mitosis is broken into these 4 parts: prophase, metaphase, anaphase, telophase, and then the whole thing is called mitosis. And then finally, at the same time that telophase is happening, we are having this J, that cytokinesis happening right there. Now, interphase, this black half circle right here, that is the place where DNA replication happens, right here in the S phase. This process that we're going to talk about is what a cell goes through every time it needs to make an exact copy of itself. That happens somewhere between 40 and 60 times in the life of the cell, depending on each cell. So remember last week we talked about how some cells replicate more than others. I actually had a couple of you guys write to me and ask me about muscle cells, and how it can be that we have all the muscle cells when we're little, when we're in the first year of life, and we don't get more as we grow up. So I wanted to give you just a little bit more detail about that. So take a look here. So it is true that most of your skeletal muscle fibers, meaning the individual muscle cells of your skeletal muscles, are formed before birth, and they don't really increase in number until after early childhood is over. Once early childhood is over. So in your first year of life, you will continue gaining muscle cells. After that, you have what you have, and they just get bigger. So you can see in this picture, this is what's called muscular or muscle hypertrophy. So this guy is the same as this guy. He's just increased the size of his individual muscle fibers through that process called hypertrophy. Now, we all know that muscles can repair. So how do they repair if they're damaged and, if they don't duplicate and you get more? Well, there's a certain kind of a cell on the surface of a muscle fiber. When we say fiber, we mean cell when we're talking about a muscle. So there are these cells called satellite cells, and they're a kind of a stem cell. Anybody ever heard the term stem cell before? Anybody know what that means, a stem cell? Nope. So a stem cell is a cell that has the ability to differentiate or turn into many different kinds of cells. So think of your blood cells. Your blood cells, what's produced in your bone marrow right at first is a stem cell. It can become a white blood cell, or it can become a red blood cell, or it can become any of the fragments that break up into platelets, or any of the multiple kinds of white blood cells. It's a stem cell. They call it a progenitor, meaning it's the beginning. It can differentiate into other things. So a satellite cell is a stem cell on the surface of the muscle fiber, and it can give you that extra growth, that extra repair that is needed. So when a muscle is stressed or damaged in some way through either exercise or injury, those satellite cells, they undergo mitosis. They have the ability to divide, and they can then activate or divide. They can fuse existing muscle fibers together. They can help them repair. They can help them grow. So even though you're born with a set of muscle fibers that isn't going to change throughout your life, the muscle's potential to grow and repair continues throughout your life because of these satellite cells. So they're the helpers of the muscle. But you can see even in this picture right here, this is how a muscle, would start. This is after, after exercise and adequate protein intake, you can get these individual muscle fibers to fill up. They get bigger, they get more mitochondria, they get more of the actin and myosin, those proteins that overlap one another in order to form the to generate the contraction. So that's what happens there. Anybody have any questions about that? About muscles and how you can have them all at birth? It doesn't look like it, does it? When you look at a 1-year-old and then you compare it to a full-grown adult, you're like, you can't possibly have all the same number of muscle fibers, but it's true, you do. All right, back to nucleic acids. So these are these large biomolecules, and they're essential to all known forms of life. All prokaryotic and all eukaryotic cells have these nucleic acids in them. Now, remember that we have our eukaryotic cell here. That's the one that has the nucleus, has the membrane-bound organelles, and then we have the prokaryotic cell without any membrane-bound organelles, and the nucleus is missing. So it has the nucleoid area right there. So inside every eukaryotic cell, there's this nucleus. Inside the nucleus, there's DNA. DNA has one major job. What's the most important thing that DNA does? I've said it before. Toby.
It gives the instructions on how to make things.
What things?
Cells?
Right.
Cells?
Lower down the hierarchy of that. Kate.
Is it proteins?
Yeah, you're on the right track there, Toby, because cells, of course, are made up of proteins. They're primarily made of protein. So that's what DNA's job is. DNA holds the code to make protein. You can kind of stop there. They hold the code to make protein. Does DNA do other things? Yes, but the very 99% importance of its job is making protein or holding the code to make protein. And proteins do almost every job in a cell, and living things are made of cells. So you guys have to understand the critical importance of protein. Keep in mind how important it is to make protein right. You'll understand how important this process is. We have to make the DNA has to be right. The copy of the DNA has to be right, or the protein won't be. And the proteins do the jobs of the cell. So to learn how proteins are made and how cells divide into more cells to make in that S synthesis phase, we have to start by knowing what the structure of DNA is. So what does DNA stand for? Reagan.
Deoxyribonucleic acid.
Excellent. Deoxyribonucleic acid. Anybody know what the shape, this twisted ladder shape of the DNA molecule is called? Kate?
Double helix.
It's called a double helix. So this becomes important when we start learning about RNA, because RNA is a single helix. Double helix means it has two of those lines on the side, the backbones. RNA only has one of those, so it'll be a single helix. But we're talking about DNA right now. So if we were to unwind this piece of DNA and lay it flat on a table, Each half of it would be called a strand. So this is a strand here, and this is a strand here. And each strand is made up of this blue, the blue squares right there represent a sugar phosphate backbone. And then all of these nitrogenous bases. So you can think of the nitrogenous bases as puzzle pieces that fit together. You know how when you're making a puzzle and you try to push in one that doesn't fit, it won't go? They have to be the right fit. Well, that's true here too. And the only thing that A will fit with is T. The only thing that C will fit with is G. They have to fit together. Now, the sugar phosphate backbone right there, that's the same molecule all the way along. We look at it here, the molecules right here. There's 1, 2, 3. They're the same, no matter what the organism is. All the DNA that you have the same sugar phosphate backbone molecules all in a row. It's only these nucleotides in here that are different. Once again, this right here is what gives you all the different cells types in your body, all the different tissues, all the different organisms that there are on planet Earth are all because of a different order of these 4 letters. So right here, you have one sugar phosphate attached to its one nucleotide. Right there. This is the nucleotide right there, or the nitrogenous base right there. So this one has an A in it. It stands for adenine. This one has a T. It stands for thymine. C stands for cytosine, and G stands for guanine. All right, now this picture shows the structure of a DNA molecule. So I'm going to turn this up, turn this around. We're going to look at this. So right here, you have your X-shaped chromosome. You can see it in 3D right there, how if we unwind it, it's wrapped around those little pink balls. Those are called histones. Now we unwind it even more, and we have this part right here. The blue is the sugar phosphate backbone, and the colors there are the bases. And each color stands for a different base, but there's only 4 colors. There's adenine, guanine, cytosine, and thymine. And here you can see them again. The nucleotides, that individual thing right there with the sugar, the phosphate, and that nitrogenous base, those are what DNA is made of. That's what RNA is made of. So the nucleotides that are found in DNA and RNA have 3 parts to them. They have this 5-carbon sugar right here. Now in DNA, the sugar is called deoxyribose. That's where the D comes from. They also have a phosphate group right here. The P with an O, that's a phosphate group. And they have some kind of base. It could be adenine, guanine, cytosine, or thymine. So the D is for deoxyribose. If we look right here, you can see a little bit more of a, of a detailed picture. I told you guys ATP is another nucleic acid. We talked about this before. We talked about what ATP looks like when we have these 3 phosphate groups, and we break this off right here to get the energy out of the ATP molecule. We've got a very similar structure here. So hopefully this looks familiar to you. So nucleic acids are storage molecules in a cell. ATP stores energy. DNA and RNA store genetic information. Right? All right, back to this picture here now. See how the differences in the rungs of the ladder are the only thing that's different about the different parts of the molecule? Sugar phosphate base is identical all the way along. So let's look at these individual nucleotides. Right there, you can see how there's 4 different colors and 4 different letters. They correspond to the color that's on the letter. So right here we have the 4 that we have: adenine, guanine, cytosine, and thymine. Now, these are the same 4 in all DNA of all organisms on planet Earth. We're using the same 4 nucleotides. Once again, kind of blows your mind when you think about all the variety that we have and how it all comes down to these 4 letters. Okay, so we've got our DNA. We know what it looks like. We know what it looks like when we unwind it. There are 2 times that we need to get a copy of that DNA. The first is in the cell cycle, when the cell needs to go through mitosis and make 2 identical copies. The second is when we need to make a protein. So we're going to talk about the cell cycle first, and then we'll talk about making proteins. Same process happens. So we'll take longer to go through the cell cycle, and then it'll be real fast to go through how protein is made. So we said that DNA has that ability to replicate itself. to make another copy of itself. And it does it right here in this S phase of the cell cycle. So here is our DNA right here. It's twisted into this double helix shape. It has to have a copy made of it. The reason it has to have a copy made of it is because at the end, we need to have two new cells that have identical DNA in them. So in a nutshell, DNA produces two brand new molecules, and each of them have half from the old molecule and half that's brand new. So each replicated strand that we have right here has one piece, like on the bottom there, that came from the original strand, the parent DNA, and the other one has it there. The new one is built. It is constructed inside the nucleus. Sort of like if you unzip that zipper and added new zipper teeth to each side so that at the end you had two zippers. Each of those zippers would have half from the original and half new. Right? Now, DNA replication happens in 3 steps. First, we have to unwind that DNA. Remember, it's twisted into that double helix? We have to unwind it. Then we have to join those pieces together. Well, we have to base pair it first. So we have to put these pieces in, and then we have to join them together, lock them together. So think of it this way. You have to unzip your zipper, put in new teeth, and then snap those teeth in place. Those are the 3 steps that have to happen. So during unwinding, enzymes are everywhere in the body, guys. This is another example of enzymes at work. Whenever you see ASE at the name of a chemical, it's an enzyme. So during unwinding, you have DNA helicase. Not topoisomerase. So DNA helicase. Somebody find it for me. There we go. I knew it was there. I was looking on the other end. Helicase. So DNA helicase is an enzyme that will open that up. But have you guys ever tried to untangle a ball of string? You know how it can get all knotted up? Well, fortunately, there's this enzyme right here called topoisomerase that separates it before. It separates it before it is split open like this so that it doesn't tangle. So it basically uncoils the DNA as the helicase opens up those strands. That keeps it from knotting up and lets that replication process happen orderly. Now, once the topoisomerase is untwisted, and then the helicase has separated all these nucleotides from one another, we got to find their matches to go here. Remember I said that adenine only bonds with thymine, and cytosine only bonds with guanine? Well, if you have here a loose nucleotide, it really wants to bond with its pair. If there's one sitting around, let's say this is adenine right here. If there's a thymine anywhere nearby, they're going to bond with one another because they really want to. They really want to form that new connection. They don't like being unbonded. Well, the nucleus is full of them. It's full of loose nucleotides. All they have to do is separate this and they immediately bond with one another. So topoisomerase is untwisting. Helicase is unzipping. You know how when you do a zipper, the 2 sides of zipper, you have that little straight metal piece that gets the zipper started. At the bottom, you have to slide it into that metal piece. Well, in the same way, DNA needs the same kind of thing. It needs what's called a primer in order to get the whole process started, just like your zipper on your jacket. So once the primer is in place, the primer would be, would start at the ends here. Once the primer is in place, they're colored in this picture right here, then another enzyme called DNA polymerase comes along, and it picks up, it's right here, this purple square. It's going to pick up those loose nucleotides that are just floating around in the nucleus, and it's going to stick them onto this strand, just like you were adding new teeth to a zipper. Right here. So this is where things get a little bit more complicated when we're talking about the adding of those new nucleotides. You have what's called a 3' end and a 5' end on a strand of DNA. Now, DNA polymerase can only add those nucleotides counting down from the 5' end down toward the 3. On the other side, the other way. So it gets a little bit tricky. Now, on the strand that is this one right here, on this one right here, from the 5' end to the 3' end, that's pretty easy because it goes in a row. We're going in this way. So it can just keep adding them here and here and here and here, working this way. But on this one, it has to add them from the 5' to the 3'. So they have to be put on like a couple at a time. It'll put on a few at a time, and then it'll go back here and put on a few more, and then go here and put on a few more. Those new pieces that are added right there, like a few at a time, those are called Okazaki fragments. So the bottom one is called the leading strand. The DNA polymerase on it just slides along its merry way. It's going in the right order. This one up here on the lagging strand, it has to jump and then go backward, and then jump and then go backward, and then jump and then go backward. Amazingly, though, they happen at the same rate. That's pretty cool. All right, I'm going to skip on past that. All right, so trying to decide. I want to make sure we have enough time here. All right, so the whole process. Let's talk about the whole process of how DNA makes another copy of itself. First, helicase unzips the DNA, right? Topoisomerase is unwinding it. Helicase is unzipping the two sides right there. That reveals these two open strands with their bases exposed right here. Once those bases are exposed, the primer is added. That primer then makes the DNA polymerase get to work. The DNA polymerase is going to move from the 5' to the 3' end, from this way to this way. On this one, it has to work backwards toward, as it continues on toward what's called the replication fork, which is this opening right here where it opens up. So as it goes, this DNA polymerase right here, this blue glob right there, moving along there, it checks its work. If it finds that something tried to get in where it doesn't fit, oh, look, a cytosine tried to bond with an adenine. That's not going to work. It takes off that cytosine and puts on a thymine. It puts on the right thing. So it checks its work as it goes. And then DNA molecules are formed, half from the original parent strand and half brand new. Toby.
How does it know, like, if it doesn't fit right?
You know, one of my favorite questions about biology is how does it know? Because there is so much about this that we don't know. It's, first of all, it's a chemical bond that happens. I'm going to back up to this picture right here. There we go. So this is a chemical bond between these. If you take a look right here, this is a bond. This is a triple bond. So cytosine and guanine together form a triple bond. Thymine and adenine, that's only a double bond that they form right there. So if you try to stick a thymine in where that cytosine goes, something's wrong. The chemical doesn't line up, so to speak, and the DNA polymerase is going to recognize that. There's something about the DNA polymerase that lets it recognize that. Now, the next logical question that would follow is, well, then how come this sometimes goes wrong? If they can only bond this way, and DNA polymerase checks it, how can we end up with mutations anyway? How can we get ones that, because that's what a mutation is one of these in the wrong place. We'll talk more about those next week. We talk about mutations, but it's really, first of all, I think it's amazing that it happens right most of the time. And then second, I think it's kind of amazing that these mutations can happen at all. So there's ways, but it's really easy for me to personify all of this and make it like, oh, the DNA makes decisions and it doesn't like the way that looks. And I use words like that. Like I say, oh, they really want to bond, like as if they have consciousness and they want to. It's all chemical. The whole thing is all just a bunch of chemical reactions that are all happening inside you all at the same time. Pretty crazy. All right, so this is what those individual nitrogenous bases look like. When we're talking about a molecule, remember what we mean when we say molecule. We just mean atoms that are bonded together. That's what a molecule is. So DNA is a molecule, and it's made up of sugar molecules that are bonded to phosphate molecules and nitrogenous base molecules. That's what we have with DNA. And going back to this again here, you can see that these two they have two rings. This would be carbon. Whenever you see ring structures like this, it's carbon. Carbon's really unique in its ability to form these ring structures. there is a lot of energy that is locked up in a carbon ring. This is all part of the reason that carbon is such a, like we're carbon-based life forms. It is the basis of life on Earth. So yeah, these two only have one of the carbon rings on them. That's another way that the DNA polymerase would recognize if it's supposed to be these two bonding together, or rather these two bonding together, and now you've got another one that matches one of those. It would identify those. All right, so here is another nucleotide right here in this red square. So see the 5 carbon sugar right here. Where we've got a circle like that, that's a sugar. Often that's a sugar. So our carbon-based organism. So we've got this 5 carbon sugar. We've got a phosphate group right here. We've got the nitrogenous base. So see that G? The G stands for guanine. All right. Now the sugar and the phosphate group in each nucleotide create this blue background right there. The sugar phosphate, that's the blue represented here. They're going to be pointing to the outside. That's the outer part of the ladder. The rungs of the ladder are going to be these colored ones on the inside. The bases point in. All right. Now, once again, remember that 3' and 5' end, because that's important in the way it has to duplicate that. All right. So these bonds right here that are in there, right here, you've got these 3 bonds and these 2. Those are called hydrogen bonds. Anybody heard of a hydrogen bond before? So hydrogen bonds are the bonds that hold one water molecule to another water molecule. I always call them the hydrogen bonds of friendship because it's like they're two molecules that are holding hands. So there'd be a one water molecule and another water molecule, and they're holding hands, and all you have to do is put a little energy in it and you break them apart. Hydrogen bonds are actually pretty easy to break, and that's a good thing because all of these are hydrogen bonds right there, and those are what are broken by that helicase. It breaks those bonds open. So the sides are the same. The nitrogenous bases are different. Since the bases pair in a certain way, we call the rungs of the ladder the base pairs. So these are base pairs right here. Now, the right side is going to be complementary to the left. That means if I know what this left side is, I can know what this side is. I don't have to wonder. If I know this is an A, what's the top? What's this one here? If this is an A, what does A bond with? Hey.
Thymine?
Yep, thymine, T. So adenine, thymine, guanine, cytosine. You can abbreviate them A, C, T, and G for this. And they only are going to bond with one another. They're going to make a complementary pair. So if this is a G, what's the next one? Hey.
C?
Yep, C. And we can continue all the way down. T, if T is loose in the nucleus and it's not bonded to anything, it really wants to bond with an A. So if there's an A nearby, it's going to bond with it. C to G, A to T, T to A, G to C, A to T, all the way along, right? If this strand of DNA right here that you're looking at here has a complementary strand, what's it going to be? What's this one going to be? Kate, why don't you go all the way down for us?
GAACATGTTA.
Very good. Very good. Now, knowing that, you can understand how proteins are made. And that's what we're ready to move into. So I told you that the first part, making another copy of DNA, was going to be the longest part of it. The same exact thing happens with a protein, but instead of making an identical copy that has 2 new strands, one from the old and one from the new, we're making a one-stranded RNA molecule. DNA makes a copy of itself in the S phase of that cell cycle. When a protein needs to be made, we have to have a new molecule altogether, and that molecule is called RNA. So remember, DNA's one job is to hold the code to make the protein, and protein is really critical. It is so important that the cell very carefully protects that DNA inside, like think of it as a fortress of the nucleus. It never lets that DNA come out of the nucleus. think about it this way. If you had a really important document, your life depended on this document, you'd put it in a vault and keep it really safe, right? If you had to show it to somebody, you wouldn't carry it around town in your pocket. You'd make a copy of it. You'd leave the original in the vault and you'd take the copy with you. That's just what DNA does. It makes a copy of itself. And the copy that it makes is called messenger RNA because it's the RNA that carries the message. Now, RNA is also really important. It has several different jobs, but they're all related to making protein. So we talked about how DNA is this double helix, twisted ladder. Look at the RNA, single helix. It has one of those sugar phosphate strands. And look at the color difference here. That shows us that the sugar is different. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. Very same. It just has oxygen taken off of it. You can also see that instead of thymine, adenine, guanine, cytosine, and thymine, RNA has adenine, guanine, cytosine, and oh, that's different, uracil. Right now, here's how I remember. You guys, I have funny ways to try to remember. They help me though. So if you have a good way, use your own. But I always remember that the letter A and the letter T make a word, at. They're the only combination of these four that make a word. So A and T bond together. If I can remember that A and T make a word and they bond together, I can remember then that C and G are what's left, and they bond. Same thing here. I think of AU as being a word. Aw. So, aw, that makes a word. Adenine bonds with uracil. Guanine and cytosine are still going to bond with one another. All right. Now, one of the four bases in DNA's nucleic acid is this thymine. The thymine is so that the DNA repair enzymes will recognize thymine rather than uracil, because thymine is resistant to mutations in a way that uracil is not. That actually makes your genetic code more durable, less likely to have mutations, and it allows it to store all the data that's needed in order for life to function. So the chemical breakdown of cytosine right here results in the production of uracil in DNA. Just that's a side note. So the result of all that, the result of everything that I just said is that DNA is pretty easy to fix. DNA can be repaired and is easily repaired. RNA is pretty short-lived. It doesn't live for very long. Any fault, any problem in the RNA doesn't really matter to the cell because it just makes more. But that DNA, that's a once and done. So it's got to be a lot stronger. All right. One other difference that you can see here is that the strand, oh, I said that right. The strand is different because it's a different sugar. So here's the two sugars. We've got deoxyribose, that's in RNA, DNA, and then ribose is in RNA. All right, so you can see why it's called deoxy. It's had an oxygen taken off right there. That's the only difference in these two molecules. But that one little difference, taking off that oxygen, deoxygenating that ribose, makes a big difference in the way it behaves. So still has 5 carbons. So ribonucleic acid, it's still going to be a 5-carbon sugar that it has. Another important difference between these two, DNA can replicate itself. We just learned how that happens. RNA can't though. RNA has to be made from a DNA molecule. Now, sometimes this RNA can loop back on itself. It can form a strand that looks kind of like this. So these are loose, remember. So sometimes it can twist back on and bind this way. That becomes really important. One of the questions that I have that I've never found an answer for is why doesn't it do that all the time? Once you've got all these loose, how do they know only to bond in a certain way? But they do. They do. And the bases are still going to match up the same way. A is going to match up to a U, and C is going to match up to a G right there in the same way. So DNA, 2-stranded molecule, deoxyribose, adenine, guanine, cytosine, thymine, right? RNA, single-stranded molecule, ribose is the sugar, adenine, guanine, cytosine, and uracil are the different ones. Now, we said there's several different kinds of RNA. They all do jobs that have to do with making protein. These are the 3 big ones. The messenger RNA comes out of the nucleus carrying the message from the DNA. The ribosome is made up of ribosomal RNA and protein. And then this transfer RNA, that's where it loops back over itself and binds in this really cool way. So let's look closely at two of them, at the messenger RNA and the transfer RNA. So this is the one that comes out of the nucleus. It is the messenger that brings the message out of the nucleus. So remember that the organelle in a cell that makes protein is the ribosome. Messenger RNA tells it what protein to make. It carries the code from the DNA to the ribosome. Now, this is one of those RNA molecules that folds back on itself. So remember that what we make proteins out of are amino acids. Transfer RNA is going to carry the amino acid. That's one right there. It's going to carry that amino acid to the ribosome. So see this AAG at the bottom? That AAG at the bottom is going to bond with a UC on the messenger RNA. And it's going to tell the ribosome what order to put those amino acids on. All right, so just for a second, let's come back to the cell cycle and making new DNA. So remember the word transcription. Have you ever heard of the word transcription before? If you transcribe something, it means you make a copy of it, right? In genetics, it means you make a copy of the genetic information. You make a copy of the DNA. You transcribe it. Now, during transcription, we have a messenger RNA copy being made. We transcribe before the next step, which is called translation. So at the end of transcription, we have one copy of this DNA that is called messenger RNA. Then transcription is the next step. Sorry, skip that one, but run out of time. Translation comes after transcription. To translate something needs to put it into another language, right? So it's basically taking the language on the messenger RNA and translating it into a protein. So we translate the language of RNA into that protein. That's the process in which the ribosomes that are either in the cytoplasm or on that endoplasmic reticulum make protein based on the code that is on that messenger RNA molecule. We call this whole process gene expression. After transcription, the messenger RNA goes to the ribosome where protein is made. The whole thing is called, in the process, called translation. All right, let me see here. I think we are going to stop there. I want to show you one quick thing. So we'll end with this video about the transcription, since I just talked about transcription. Watch this.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
So this is another one of those, how does it know? How does it know what to remove? How does it know what to process? We know that it does. We're not exactly sure how it does, how it knows to do that. Pretty cool. All right. We'll pick up right here next week, I think. I gotta see how we're doing. All right. Have a good week, everybody. Good to see you. Bye.